Hi everyone, my name is Christoph, and today I'm going to show you in this video how to use this MuJoCo HAPTIX spike train analysis pipeline. This pipeline has three components for it. The first one is sensing data using MuJoCo HAPTIX. The second part is a pre-processing stage that happens primarily in MATLAB. And the third part happens with Python tools used for analysis on the data that's been sensed with MuJoCo. This workflow can be used as one full pipeline, or you can use these three components independently. Today in this video, I'm going to show you how to use them all together, and that functionality is captured in this notebook. Let's get started. So first things first, there are a couple dependencies that are required before getting started here. One of the major ones is MATLAB. MATLAB is used to control the hand model in MuJoCo HAPTIX, the little hand model that you see right above me. It's also used for pre-processing, which turns the data that is captured from MuJoCo into a format that's more familiar with spike train analysis. That's using a tool called TouchSim. And lastly, there's a MATLAB API for Python that's used in this Jupyter notebook that controls everything and lets it all run all together in one simple key press. So first things up here is that you're going to have some imports that you need to handle. This is going to be good because everything should be captured inside of the repo. So one quick run of this, you should be able to have all your tools imported. And if you have this working, then everything else after it should be good to go. Next, in this second code cell, you want to have 3 directories specified if you have intentions of running this full thing. If you're just running a component of the pipeline, then only a couple of them have to be specified, but you want to make sure that you have these specified so that you're not stepping on other data that you might already have. The first one here is going to be a save directory for where you're storing data sensed from MuJoCo. This hand model above me is going to rub the little key object right in front of it, which is going to produce these sensor values. This is the first directory here is the one that you want to save those values to. The second one is after the pre-processing step that we use with the MuJoCo data to create a neural response. It's basically converting the data to another format. That's where you want the second directory to go to. I call that spikes in this one. And the third one is if you're using TouchSim to convert that neural response data into a Python-friendly struct format, then you can have an additional directory specified if you want to work with that data using Python. So next up, you want to call this MuJoCo sense function. It's a class constructor, which is basically going to spawn up in a MATLAB engine in the Python instance that's running right now. And what that will allow you to do is you can use Python to call MATLAB statements. The ones I'll be using for this demonstration have already been predefined and pre-written. They're in the MATLAB files inside of the repo for this project. But this can be modified such that you can call any additional MATLAB functions that you may need to leverage. One quick mention as well, this function right here is going to go through the entire MATLAB path and import every single directory or file one by one. So this function can take a couple minutes to run. Depending on the project that you might be working with one day, you could import a subset and still have the same functionality. But to make sure that this is transferable and works on different machines, it imports everything. So that's going to take a couple minutes here. So now all MATLAB paths are imported, which means that I can now use Python to call any MATLAB functions that are reachable through my MATLAB path, which is really good. So next up here, the first thing that you'll see is this line that's called mj.eng.mjclose. That's a function in the MATLAB path, which basically cuts out the connection with an existing instance of MuJoCo, the hand right above me, just in case one was already running from previously. It's there so that in case an error pops up saying you're already connected, this function will terminate that connection so that you can start up a new sensing session. The next function here is mj.hapticsense, which is basically a Python API call to a MATLAB script that will take the hand above me, and right now it's specified for 10 times to swipe along that key object, which basically generates 10 different trials of data. And so if you walk through the parameters right here, the first one is spikes path. That's a directory that we specified where the data is going to be saved. The step count is essentially going to be how many steps to take for the full swipe. I have 1,000 specified right now, so there are going to be 1,000 increments to move that hand along the key object. The third one is going to be a target sampling frequency, which basically in MATLAB there's a timer that's running in the background which seeks to capture a sense measurement on a given interval. And so with a target sampling frequency of 100, this is saying that I want to capture 100 sensor readings every second. The reason that this is important to know here is because if you change up the step count, it'll take less increments or more increments to do the full motion across the key object. Depending on how many increments you have, this can be a longer or shorter process. And so if it's taking longer, you may have an interest in using a higher sampling frequency or perhaps even a slower sampling frequency depending on what you're trying to observe here. And the last parameter is trial count. That one is currently specified to 10, and it means I will generate 10 trials or do 10 swipes one after another before moving on in the code. And so if I run this, it's going to orient the hand right above me and begin to swipe that key object 10 times. When this MuJoCo simulation is swiping along that hand, this fingertip that's touching the key object right there is going to encounter sensor measurements, which are going to be forces in newtons. This force in newtons can be used and converted to a value in millimeters to correspond to a depth of indentation that that finger is experiencing. When we have that value, that allows us to then further create the neural responses because an indentation depth in millimeters is required as an input to that. And so that gives us a neural response that we can use for further analysis. When that's finished, you can see that it gets saved to the directory that you specified earlier, which will then allow us to move on to this mj.mujoco to spikes function. The only parameters that you need here are a data path to input, which is going to be where that MuJoCo sensor data was saved. And then you'll also need a save path to save the pre-processed data to. This output data is going to be primarily in a MATLAB format, so if you want to perform analyses on it in MATLAB and use the tools there to work with the data, perfectly fine. Once you're done with this function, you're ready to go. Otherwise, you can continue working through this workflow and convert it to the Python-friendly format to use some of the spike train analysis tools that are also included in this repo. So the pre-processing step just finished, and before we continue, I'd like to show you here, this indentation depths folder is where those initial sensor values from MuJoCo were captured that the hand model up there was generating. That's sort of this indentation depths, and we can see the file format structure. Object corresponds to the key object because that's what MuJoCo is using here. The sampling frequency is 416 for this first trial and ranges about 416 up to about 473 across all 10 trials. And then you have at the end the trial that's specified so you can go back and see in order how things happened. If we back up here, we can go into the spikes folder. Now we should see all the pre-processed files. That's where it says spikes to correspond with the neural spiking activity that we use that indentation depth to create the now neural responses throughout the entire fingertip. The last step here is going to be this TouchSim conversion to a Python-friendly format. We'll run this last notebook cell to kickstart that process. This one should take a little bit less time. And what that is going to allow us to do is then open up these MATLAB files or the data from the MATLAB files in Python and then use some visualization tools that are customly built for spike train analysis. Awesome, so now that's done. So now, if we want to continue with the pipeline, we're ready to perform some spike train analysis in Python. The first cell to get that process started, we're going to hard code the files that we need for performing the analysis on. Or alternatively, there's a trial select function that comes from the trial stats Python file, which will automatically allow you to select files that match a particular criteria. So for example, here, I want to grab every single file that corresponds to object key. Object key is what I use to distinguish MuJoCo away from data captured by sensing other objects. And also the spikes directory here is just going to be what top level directory do I want to begin searching from in order to find the files necessary. The inner workings of this function are going to be detailed in a different section. In this notebook, there are 2 examples for spike train analysis. There are a couple other ones if you want to study neurons from a different perspective, but these are some examples to help get you started to see how exactly this component of the pipeline works. The first one here is going to be comparing afferents, and so the comparison here is going to be between slowly adapting neurons and rapidly adapting neurons. What this means is that from the 10 trials I just selected from, because all 10 of them match this object key, I'm going to take every single neuron response that is from a slowly adapting neuron and all of the neuron responses that are from a rapidly adapting neuron, and then create a probability distribution of both of those independently to show the likelihood of spiking given this interspike interval. And so if we look here, we'll run this since we're working with brand new data. It's going to take a second to load up the data from each file so you can actually see in real time, are the files being loaded the ones that you intended to select from. And once that's done, this pulled from all 10 files all of the data from slowly adapting neurons. And I'll do the same thing for the rapidly adapting neurons right here as well. And we'll see that there will be two unique probability distributions. And because they're different, we can compare the two using a KL divergence value, which we see here is 0.6426. So we can see a little bit of similarity in that it is right skewed. The slowly adapting neurons tend to drop off significantly faster than the rapidly adapting neurons. And another comparison we can run as well is by comparing trials. So if you wanted to compare specifically trial 1 versus trial 2, the key object presents a relatively simple experiment, but for more complex interactions with MuJoCo or a physical hand model, you may be interested in comparing trials to see if there's any intricate differences between those two trials. We specifically said that we want afferent type rapidly adapting. So this is going to pull all the rapidly adapting neurons from trial 5. This alternatively could be all the PC neurons or all the SA neurons or just all the neurons all together. And rather we could specify a specific neuron, or depending on the title of the MAT file, we could specify what dimension along the key object that we wanted to grasp, or we could even look at the distance away from the stimulation point to make a probability distribution. There's a lot of different ways to go about these analyses. And so depending on what you're interested in finding, chances are there's a good way that you can approach it. And so we can see this time that again, we have two different distributions. Trial 2 was a little bit different than trial 5, but they're a bit similar because we ran the exact same experiment. So we should see a lower KL divergence. We see 0.1509 this time, which is lower than the one that we saw earlier, 0.64. And so this makes sense that the same experiment should generate relatively similar outputs, but not exactly identical just because they didn't occur at the exact same instance. So that's how you use the full pipeline. Again, this whole pipeline exists in 3 individual components that can be used separately if it better suits your needs. What I just showed you, however, is everything from sensing the data using MuJoCo right above, to pre-processing it, and also generating the visualizations. A next step to go forward from here could be developing a classification tool, for example. But that's all I have to show for you in this video. I hope you learned something useful. I hope you learned something new, and best of luck to you in your analyses. Thanks for watching.